Hello, everybody. It's lovely to be here. I'm Brian McGee. I'm the CBO at a company called Anocca. Before I get into telling you a little bit more about who we are and what we're up to, I just want to say it's great to be at Meeting on the Med. This is my first one. It's a lovely conference, really intimate, very friendly, and I think it's nice to be surrounded by so many people who are, I guess, optimistic and fighting the good fight against some of the worst diseases that you can imagine. I think we're going to turn the tide, and I'm super excited to see what comes and what comes next. That's part of the reason why I'm at Anocca, so let me tell you about us. We are Sweden's largest and fastest growing privately held biotech. We've raised $125 million to date, and that's to develop TCR-T-based immunotherapies. But we're a little bit different from, I think, a lot of other folks in the TCR-T space in that the first thing that we've invested in doing is building a really unique industrial cell-based discovery platform. And that allows us to, with extremely high levels of precision and speed, reproducibility, accuracy, et cetera, build libraries of TCR-T therapeutics. And they have deep coverage of two things. One is HLAs, which is, I think, for those who aren't in the space, a way of thinking of different patient segments. And they also cover lots of different cancer targets. So our vision as a company really is to make most cancers treatable in the vast majority of patients. It's exciting times for us. We're teeing up and getting ready to go into clinic in 2024 with a KRAS TCR-T. That's going to go after pancreatic cancer. So I think we might get bravery points. I think in some quarters we might get a bit of quizzical looks, but this could be a big deal for patients, could be a big deal for us. It's a big deal for the field, and so we're super excited about the next 18 months. I think it's worth at this point just taking a little bit of a step back. I had a question yesterday when I was in a conversation where someone said, why don't you just make a CAR against KRAS? And it's actually a more common question than you imagine, and it's not silly at all. CARs are incredible. CAR-Ts really revolutionized the field. It's ushered in, I guess, an age of what feels almost like science fiction, unimaginably complex therapeutics that have phenomenal impact on patients. And the kicker with CAR-T is that they're really limited to a fairly small number of cell surface targets. The 400 or so that are in clinical development today, really they're going after 19 targets. And that's just, at the end, when it comes down to the commercials of these assets, it just won't work. There can only be a very small number of winners in that game. We see TCRs as the next obvious targeting moiety that you can strap on to a T cell. And the benefit of TCR is that it accesses the entire cellular proteome. So you can engineer and tune a TCR to go after almost anything that you imagine or anything that you want. And the even better news is, when it comes to cancer specifically, we've been characterizing cancer for such a long time that there's an abundance, an absolute wealth of different available cancer targets that you can go after. So it's a very winnable game, and it sounds like TCR-T is a bit of a magic bullet. The reality is it gets a little bit more complicated. When you zoom into that little cartoon, you've got three specific, naturally I call them problems, right, because I'm a pessimist at heart. But you've got these specific challenges you need to address and manage. The first of them is the HLA challenge, which I alluded to earlier. It's a way of thinking of patient segments. If you're extremely pessimistic, you start with the statistic that there are 13,000 different HLA variants. When you do a bit more complex maths, you get into the kind of slightly more optimistic picture that with 20 HLAs, you cover upwards of 90% of the world's population. So this is a winnable game. It's fairly manageable. You can even get to in the 80s with 10 HLAs. And these things present antigens. So you start compounding the complexity now, right? If you've got a fairly big protein inside a cell and a HLA, you'll start seeing it cut up different and presenting different pieces of antigen target. So what you're really in the business of doing in the TCR space is finding targets within targets. And then you reach the TCR challenge. You now have to find a targeting TCR that targets the target in the target. And I'm already tongue-twisted with saying the word target. But What's important about that TCR is not just that it recognizes that HLA antigen complex, it's that it doesn't recognize and go after anything else. That's pretty fundamental, because you start having horrible conversations where someone says, if we get this wrong, we melt someone, and that is not something we want to see happen. So Anocca really, I think at this stage, we've operationalized a platform that tames a lot of this complexity, and we base it on 3 components. And the first thing that we do is fairly different from a lot of others. We don't really rely on anything algorithmic. We don't use AI machine learning to predict targets. We use real cell biology. And we think that's really important to us, that you do this in the context of actual cells. To do that well, and getting into the maths that I kind of notionally talked about with HLAs and antigens, you've got to do a lot of these experiments, so you've got to do it industrially. This is our belief philosophy, right? Like, you can disagree with me. We do real cellular experiments at scale, reproducibly. We do it quickly, and we do it cheaply. Cheaply is often a word that's left out because it feels vulgar in the space, but we do it quickly and cheaply. We use software to accelerate that and manage the workflows because it gets complicated really quickly. When they founded the company, they realized there wasn't any off-the-shelf software that was suitable, so they had to build it, and now 10% of our team are software engineers. So we're very committed to that path. What this allows us to do is a kind of end-to-end discovery flow that goes from a target of interest to an actionable TCR-T inside 12 months. And it starts with antigen discovery. I think antigen discovery is one of these things that's just seen as easy in the space. And I think we would assert that it's not so straightforward, because it's important that you can look at these antigens in an individualized HLA. We engineer a monoallelic HLA cell line. We push a protein into that of interest, and then we map it. And we can do that quite extensively, which lets us know what's the best antigen target to go after in that space. And that's important because we're going for quality from day one. We're not making any guesswork or assumptions. We just don't think that the AI or algorithms are just there yet. We then step into using that HLA antigen complex that we've identified, the best one that we've selected, to go fish for TCRs. We do that from healthy donors, but for various reasons, but one of them being they've been through thymic selection, so you can infer a degree of safety in the TCR. At that point, we validate them. So we say, hey, these things work in a tube, that's great, but let's make sure that we can put them back into cells and that there's an interaction there, a real biologically valid interaction. I mentioned safety earlier, and I'll come back to it now. While this is heavily redacted, and there are probably versions in the public domain that have lots of stuff on, but they take that out because I'm not a scientist and I trip myself up. A lot of our platform is based on recreating all the potential ways in which this could go wrong and making sure that the TCR we're advancing as a candidate doesn't have any activity on them. Alongside it, there's a pretty robust engineering protocol if we need it. Often you can find really beautiful TCRs that are just native and happy, but sometimes you want to engineer them for improved safety, improved potency, or both, at which point you go back through that cycle of safety to make sure that they are good and trustworthy. But it just helps us get around the limits of the repertoire. I mean, I'm not an immunologist by background. I'm a plant scientist originally, and so this stuff is insanely complicated to me. Plants are wonderful because you can do whatever you like to them and they kind of survive. Humans are very different. But I find it fascinating how an immune system has evolved to find things that it's, how do you find stuff you've never seen before? It's crazy, it's beautiful. But this kind of flow allows us to build a pretty extensive pipeline. It's all preclinical currently. And I think this chart demonstrates a few things that are relevant. I'm going to touch on PRAME. PRAME's a fairly large protein. It's more than 500 amino acids long. We can very quickly find across a whole bunch of HLAs that we don't disclose what are the best antigens that are being presented by PRAME, and then find great TCRs against them. That's easy. Well, I shouldn't say that because I'm not the scientist that worked on this, and I'm maybe not giving them sufficient credit. But you can easily cover large chunk of the population. We're getting to 85% of the US and 95% of the Japanese population with 10 TCRs. So this becomes a winnable game whereby a patient goes in, has a PRAME-positive cancer, and isn't then faced with the reality that, oh yeah, that treatment you read about, sadly not available because you're not an A2 HLA person, bad luck. It doesn't work quite as well with KRAS, although I think when you look at the field, we're able to expand that beyond the traditional 1 or 2 assets that folks have. But the challenge with KRAS is that it's a single amino acid mutation, so it's inherently a lot more challenging to raise multiple TCRs in any HLA background. We're continuing to work on this, but we think we're getting into an exhaustive space with that coverage of the population. But really, the platform allows us to offer a library that radically expands the commercial and clinical utility of TCRT. And the vision for this really is that, I guess I've alluded to some of this already. You have coverage if you've got the right HLA and if you've got the right target, but the future vision is getting to a place where you're not just deploying a singular TCRT asset, you're deploying multiple, typed against HLA, typed against target. To do this, it won't be autologous. I think it's fair to say in the future we're going to have to move into some kind of allogeneic deployment. And one of our partnering objectives when we're at conferences, my colleague Mark's in the audience somewhere, I saw him earlier. Mark's full-time obsession and job is to know everybody and know what the peer biotechs are doing with technology that can make this deployment of a library a reality. So we're super excited to meet folks who've got non-viral gene editing, allogeneic cell, iPSC stuff. We want to know all of that. We want to understand it. That's the future vision. Let's hope that we get there. I hope we get there in my lifetime, but this could be many decades away, or it could be 5 years away. We all know how biotech operates. The, I suppose, exciting point, which we're going to do with a more traditional autologous approach, is moving into a first trial in pancreatic cancer in 2024. As yet to be determined candidate set in that probe indication in pancreas. We're really hopeful that this hits in a big way. There's some clinical precedent for that. And then, as you'll see with all of these trial designs, a pretty standard intention to expand those indications into colorectal and lung and potentially others. I don't have a timer, but I guess I'm coming up on it. I just want to say, that what we've done so far at Anocca has been relatively stealthy. We've built a really nice intellectual property portfolio. We're based in Sweden, so we benefited from that legacy infrastructure of AstraZeneca. So we have a really awesome set of offices and research facility and manufacturing, which has been repurposed for cell therapies. Can't do this without an amazing team. We've built a wonderful, super international team of people, mainly scientists, with some engineers and software developers, and a couple of freeloaders like myself. But we're working very hard to, I think, change the world. And I'm super thankful to our investors who've committed a lot of money and are very patient and very committed to our vision. But as I said, Mark and I are around at the conference, and despite my face, I'm relatively friendly, so please come say hello, and thank you.